Good morning, junior techies, and welcome to this video dealing with the Quadrat method. So this is further to that first theory video where we introduced you to the different techniques in trying to determining population size. So what we're going to do now is we're going to look specifically at the Quadrat method when it comes to the indirect technique, or one of those indirect techniques in determining population size. So let's have a look here very, very quickly at what the Quadrat method is all about. Okay, so firstly, when we look at the Quadrat method, it is a sample that we're taking in a much larger habitat, which is that area in which that specific organism lives. Okay, so the Quadrat method we usually use for plant populations that don't move around too much. Well, plants don't move around too much, but populations that are pretty, they have that fixed location. They're not going to be going anywhere. And we can determine the population size using this specific method, the Quadrat method. So we're looking at determining or creating a sample area. So our sample area is an area we know, and it's usually a square because that's easy to calculate. Here we've got length times breadth, and that gives us our sample area within a habitat. Now, also important to know is the area of the habitat that we're going to be taking our samples in. And then our samples are going to be taken at random. Okay, our sample quadrats are going to be taken at random within the habitat to make sure that we have a valid investigation here in terms of determining the number of population or the number of individuals in a population in a given habitat. Okay, the actual number of individuals of a certain population can be counted within each one of these quadrats, so that's important for us as well. And if we look at insects that do move around a little bit, this becomes so difficult because there are going to be some individuals that enter and some leave in each one of these sample quadrats. So ideally, this is going to be used for our plant populations. Okay, we use our quadrats to get an average in terms of population size over a much smaller area when we compare it to the large habitat that we're going to apply this sample to. And then we're going to estimate the size of the population for that entire area that we are taking the samples within. Okay, and then once again, just here, they are extremely, well, this method is extremely accurate when it comes to plant populations or any organism that doesn't necessarily move around too much. So we're going to look here at using the formula. Now do get to know this formula because in some instances, you're given this formula in your assessment, but in others you might not be. So to get to know this formula is super important because you can maximize your results here. So for a calculation like this, you can be anything between 3 to 5 marks, and you want to make sure you capitalize on that definitely. Okay, so here we're looking at the number of individuals in a quadrat area. So this is that sample area. And then my habitat size, which I need to know. So in other words, we need to make sure we know the area of the habitat that we're taking the samples in. And we need to know what our quadrat size is. In other words, our sample size. And the units here become so important. This is one of the big points or the big areas that the students make the biggest mistakes in. They forget to make sure that the quadrat area and the habitat area are in the same unit. So measuring in square meters versus square kilometers, for example, they are not relatable units of measurement for area. So we need to make sure that we convert so that our area for quadrat size and habitat size are the same. Then we can use this formula. And then it becomes quite simple actually to apply. But let's have a look at an example here. So here you can see an individual who's sitting with one quadrat that has been placed randomly within an environment, and a sample is taken within one quadrat. Now if you have a look at the little picture on the side over there, so she might be measuring the flowers found within her quadrat. She's busy counting them there, but you can see the larger habitat area with a number of different quadrats in that area, and they're placed randomly. So random samples are taken for this area, and then we're going to look at applying the formula that you see at the top of your screen at the moment. Okay, so you can see the whole area is covered by our quadrats randomly. So here is one quadrat. This just shows you how we're going to actually be counting the individuals within the quadrat size. So you can see here that if this quadrat measured 1 meter by 1 meter, which it will, we can see that there are only 3 individuals that we can be counting, or that we are counting inside of this space, even though there's a little bit of the 4th petal that sort of is inside that space. The individual itself is not entirely inside that space, or not more than half of it is inside that space, so we don't really count it inside that space. So we need to know the area of our sample size, or our sample quadrat, and in this case I'm just giving it a random measurement of 1 meter by 1 meter, which is generally the norm when it comes to a sample quadrat when we're measuring something like flowers or insects in a larger habitat area. So looking here, to measure the area, we take the length times the breadth, and in this case it's 1 meter by 1 meter, and that gives me a sample quadrat area of 1 square meter. Okay, now we're going to be looking at the area of the habitat or the environment that we're going to be calculating the population in. So in this case, you can see that the much larger area I have as 8 meters by 8 meters, so it's a bit larger, and the area calculated here is going to be 8 by 8, which gives me a 64 square meter area in which I'm going to be taking my samples. Now, if you look carefully, you can count that we've got 10 sample quadrats. So I'm giving them each a number, very vaguely there. You can see that I've given each one a number. And this is so that we can take note of or record the area that we're finding each of these samples in. So I'm just doing the first 5 very quickly there. So my sample is taken over 10 quadrats that are each 1 square meter big, which means my entire sample is going to be taken over 10 square meters, because there are 10 quadrats spread out randomly over this entire area. Now, I've just summarized very quickly the number of individuals that I've counted in each of the first 5 quadrats, and then we're going to quickly do the next 5. And what I'm going to end up with now is a total number of individuals for those 10 quadrats. Okay, so if we count nicely, you'll see that we end up with 29 individuals in those 10 randomly placed quadrats over the much larger habitat for these plants, in this case the flowers. Okay, so where to from here? Well, now we've got to apply the data that we have to that whole 64 square meter area that we have, or that we're taking our sample within. Okay, so here we have our formula, and let's just quickly, I'm just going to reiterate very quickly that getting to know this formula is super, super important. If you know the formula, substituting into the formula becomes so easy. And you'll see I've just done it there as well. So the number of individuals in the quadrat area. Now remember, we took 10 samples, which resulted in the 10 square meter quadrat size. So you can see those values I've used. I've used my total values. The more quadrats I use as a sample will increase the accuracy of this calculation. Okay, so with every quadrat extra, I'm increasing the quadrat size as well as the number of individuals, because I am going to be counting new individuals in that new added 1 square meter space. But now I'm just putting in the values that I have for the investigation that I've done. So I've got 29 individuals in the quadrat area. The habitat size in square meters is 64, and the quadrat size, so in other words, the size, the total size over which I've taken my sample is 10 square meters. So now I do my calculation. Right, I've got 29 times 64 divided by 10, and you'll see I end up with a value 185.6. Now, let me just point this out quickly. Here, I have a 0.6 of an individual. Now, that's not really possible in the context of what we are doing here. So we are going to do away with the decimal, and we could say that our calculation has resulted in 185 whole individuals in the sample that I have taken. All right, so keep that in mind. We do not give our answer with a decimal, with a point something value. We need to give whole individuals here. So 185 would be my answer here. Right, Junior Tickies, I hope that I've helped you understand a little bit more in applying this formula, particularly in this method of determining population size. This is the Quadrat method. There is another theory video 3, that's going to deal with the mark recapture method. So make sure you get a hold of the next video, get a hold of the worksheets, the worksheet memorandums, practice those questions to make sure that you are prepared adequately for the end of the year exams. Thanks, Junior Tickies. I do hope to see you again soon.